Scientists at Scripps Research have revealed the three-dimensional structure of flycatcher 1, an aptly named protein channel that may enable Venus flytrap plants to snap shut in response to prey. The structure of flycatcher 1, published February 14 in Nature Communications, helps shed light on long-standing questions about the remarkably sensitive touch response of Venus flytraps. The structure also gives the researchers a better understanding of how similar proteins in organisms including plants and bacteria, as well as proteins in the human body with similar functions, called mechanosensitive ion channels, might operate. In response, cells then alter their behavior. A neuron might signal its neighbor, for instance. The ability for cells to sense pressure and movement is important for people's senses of touch and hearing, but also for many internal body processes, from the ability of the bladder to sense that it's full to the ability of lungs to sense how much air is being breathed. Previously, scientists had homed in on three ion channels in Venus flytraps thought to be related to the ability of the carnivorous plant to snap its leaves shut when its sensitive trigger hairs get touched. The fact that variants of this channel are found throughout evolution tells us that it must have some fundamental, important functions that have been maintained in different types of organisms, says co-first author Sebastian Jojoa Cruz, a graduate student at Scripps Research. In the new study, the researchers used cryo-electron microscopy, a cutting-edge technique that reveals the locations of atoms within a frozen protein sample, to analyze the precise arrangement of molecules that form the flycatcher 1 protein channel in Venus flytrap plants. When the team determined the structure of flycatcher 1, they found 6 linkers in the down position, and just one flipped up. The architecture of flycatcher 1's channel core was similar to other channels that have been studied for years, but these linker regions were surprising, says Kei Sao-Tome, Ph.D., a former postdoctoral research associate at Scripps Research and co-first author of the new paper. The profound effect of this mutation tells us that the conformations of these 7 linkers is likely relevant for how the channel works, says co-senior author Swetha Murthy, Ph.D., of Valam Institute at Oregon Health and Science University, a former postdoctoral research associate at Scripps Research. Now that they solved the molecular structure, the research team is planning future studies on the function of flycatcher 1 to understand how different conformations affect its function. More work is also needed to determine whether flycatcher 1 is solely responsible for the snapping shut of Venus flytrap leaves, or whether other suspected channels play complementary roles. In addition to Jojoa Cruz, Sao-Tome, Murthy, Patapotian and Ward, authors of the study, Structural Insights into the Venus Flytrap Mechanosensitive Ion Channel Flycatcher 1, Che Chun Alex Sui and Wen-Xin Li of Scripps Research, and Mark Sansom of University of Oxford. This work and the researchers involved were supported by funding from the National Institutes of Health, R01HL 143297, R01HL 143297, a Ray Thomas Edwards Foundation grant, the Wellcome Trust, grant 208361, Z17, Z, the Biotechnology and Biological Sciences Research Council, grants BB, N000145, 1 and B. Nature Communications, 2022, 13, 1, doi, 10. 1038 per second 41467, 022, 28511, 5 Scripps Research Institute. Scientists reveal how Venus fly-trap plants snap shut. The mechanosensitive ion channel is related to channels found in variety of other organisms. Science Daily. Scientists reveal how Venus fly-trap plants snap shut. The mechanosensitive ion channel is related to channels found in variety of other organisms. Science Daily. https://ift. tt. WEBJN8L. Accessed February 17, 2022. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.